The quantitative Western blot is useful in cell and molecular biology to understand changes in protein expression in a measurable way. It is important to remember, though, that the fold change or percentage change derived is a comparative number within the context of another protein and the experimental design. The fold change is not an absolute quantification of protein expression. Similarly, it's important to understand what does not constitute a quantitative Western blot. It is also important to understand how a quantitative Western blot is different from a semi-quantitative Western blot, even though, as we mentioned earlier, both terms are often used interchangeably by researchers. Here are a few examples of what does not constitute a quantitative Western blot. A simple visual analysis of protein abundance without any mathematical measurements. A lack of appropriate controls, which help optimize and validate your reagents. Detection of proteins outside the linear range, thereby misrepresenting protein abundance. Lack of in-lane normalization to correct for variability. Using imaging system detectors with low dynamic range that do not capture all of the signal present in the single blot image. All of the above will give you a semi-quantitative or a qualitative Western blot at best, but not a quantitative Western blot. Ultimately, we need to ask ourselves, why is quantitative Western blotting important and what value does it bring to your experimental study? Western blotting technique may help in testing hypotheses objectively under the assumption that experimental framework is sound and optimized. Western blotting, when done reliably and reproducibly, is a quantitative approach to study changes in protein expression and can therefore help in arriving at plausible cause and effect conclusions. Some applications of quantitative Western blots include evaluating protein expression changes under varying experimental conditions, studying protein-protein interactions, comprehensively studying signal transduction pathways, studying changes in protein modifications such as ubiquitination, phosphorylation, acetylation, etc.